Good morning, everybody.
Hey, good morning, Dennis.
Good morning, Dennis.
How are you guys doing today? Happy
Morning,
Friday.
everybody. Yeah.
Doing well. It's beautiful here in the greater Boston area. Got a nice blue sky. It is cold.
Yeah.
No snow dump today.
No snow dump.
Blue sky on the West Coast also.
All right. And with that, without further ado, let's get started for the 2024 drug approval highlights. So one of the things that I wanted to point out, I wanted to give like a little bit of an overview of the drugs that have been approved in the year before we do like a deep dive with our team into, really specific case studies. So in 2024, we saw 50 drug approvals, which is slightly down from the 55 drug approvals. So yeah, we have about 50%, we have about 60% of that being small molecule approvals. So 31 of the 50 molecules are actually, or of the 50 approvals are actually small molecules. About 48 of those, for 48% of those were actually first in class. 52% received orphan designation. 44% received smart fast track designation, and about 60 or about 36% received breakthrough therapy designation. So you're seeing by those numbers that there's really a significant amount of breakthroughs and new therapies really on the market. So I want to do a little bit of a deep dive into each of these disease areas. Now, moving on to kind of the first slide. So here in the oncology area, we saw 6 approvals in that space, and I want to go high level into each of those so that you can learn a little bit about those. So here, the first compound that is a type 2 RAF kinase inhibitor for low-grade gliomas that are harboring BRAF mutations. This is a compound, tovorafenib, which is also used in pediatric indications and it's originated here from Sunesis and Biogen and also passed through Takeda, I think, in the 2010s, but ultimately was licensed to Day One Bio and brought to the market.
Dennis,
Another really
Dennis, I see Dan Erlinson is in the audience who, Dan, I don't know if you were at Sunesis at that time, but this is quite a long journey.
Absolutely. Yeah, welcome, Dan, to the show. We also have in that same space in the glioma approvals, we have the IDH1, IDH2 inhibitor vorasidenib, which is a very interesting compound here. Actually, 70% of low-grade gliomas contain those IDH1, IDH2 mutations. And this compound has shown really remarkable efficacy in phase 3 trials, really increasing the progression-free survival by over 2 years. So previous IDH1 and IDH2 inhibitors actually were not that brain penetrant. And this compound here, vorasidenib, which was licensed by Servier in 2021, is actually brain penetrant, which is making a huge difference in that particular field. So very, very interesting discovery, originally from Agios and then licensed to Servier. Another interesting compound in the oncology space is this covalent mutant selective EGFR inhibitor. You can lazertinib. You can see this covalent warhead right here. And that is being used for metastatic non-small cell lung cancer with EGFR exon 19 deletions, and has been licensed to Janssen in 2018, and ultimately they brought it to the market. We also have inavolisib here, which is a PI3K alpha inhibitor and degrader. And I won't steal the thunder here. Medhes is going to be talking about this compound a little bit later. It's a really fascinating discovery story that Matt will share with us today. Similar for revumenib, Dennis is going to cover this right after I'm going to be done with the introduction. Very interesting compound, first in class menin MLL PPI inhibitor for AML. A super interesting story that Dennis is going to share with us a little bit later. Also, the last approval in the space, last small molecule approval in the space of oncology is ensartinib here. And ensartinib is an ALK inhibitor that is being used in ALK-positive non-small cell lung cancer. So it's rivaling compounds like crizotinib, which is also an ALK inhibitor, but also hits other tyrosine kinases like ROS1, RON, HGFR, and also this rivals lorlatinib, which is also an ALK and ROS1 inhibitor. Very interesting story that we have on the website here as well. So ensartinib actually doubled the progression-free survival in a phase 3 trial compared to crizotinib to about 2 years. And it's a compound that's been discovered and approved by Xcovery. In the genetic disease spaces, we also had 6 different approvals, very interesting approvals here, starting with this interesting HDAC inhibitor, givinostat, for Duchenne muscular disease. So Duchenne muscular disease, really a very difficult disease to treat, particularly with small molecules. There's not that much available. So typically it's treated with glucocorticoids such as prednisone. But also antisense oligonucleotides have been approved, and those are pieces of DNA that are used to mask the exon that needs to be skipped when making, in this case, the dystrophin protein. There are 2 antisense oligonucleotides approved here in the space of DMD, and only 8% of the patients can basically benefit from one and 13% from the other. So this small molecule is a very interesting approval here, comes with a hydroxamic acid, is the 6th approved HDAC inhibitor. Typically, HDAC inhibitors are approved in the space of oncology. Often they come with kind of boxed warnings for thrombocytopenia and QTc prolongations. This one here is dosed as an oral suspension, and it has shown to kind of slow the progression of DMD, reduce the inflammation, and decrease the fibrosis that's associated with the disease, as well as helping to regenerate the muscle. So very interesting approval here as well. Mavorixafor is an interesting CXCR4 antagonist for WHIM syndrome. So CXCR4 antagonists have been approved. They're most prominently plerixafor that has been used in the space of multiple myeloma for stem cell mobilization, actually. Last year, we also saw an approval in that very similar space, motixafortide, also for stem cell mobilization in multiple myeloma. So here, it's a first-in-class approval for a rare genetic disease for a CXCR4 antagonist, in this case for WHIM syndrome. And WHIM syndrome really, that basically leaves affected people here vulnerable to infection. And this therapy helps with increasing neutrophils and lymphocytes that address this particular immune dysfunction. So also, again, very interesting approval in this space. We saw 2 approvals in the space of Niemann-Pick disease, both with unknown MOA. So we saw this compound here, which I think was very interesting to have this N-oxide here, as well as this somewhat chloroxime functional group here. So Niemann-Pick disease, to talk about this a little bit, is a rare inherited metabolic disorder that's also called lipid storage disease. And it leads to kind of abnormal accumulation of lipids, for example, sphingomyelin or cholesterol in various tissues, including the brain. So the type C is actually affecting the brain a lot. Again, the other compound that was approved for a similar indication here is just the derivative of the amino acid leucine. You can see that easily here. It's an acetylated version of the amino acid leucine. Another interesting approval, again, in the space of rare and genetic diseases is this CRF1 receptor antagonist for the treatment of congenital adrenal hypoplasia. And that is a disease that's affecting adrenal glands, leading typically to a deficiency of key hormones like cortisol, but an excess of androgens. And most commonly in this disease, in kind of 95% of the cases, 21-hydroxylase, which is a key enzyme in the biosynthesis of cortisol, is not functional. So the CRF1 receptor agonism reduces basically the action of a specific hormone and thereby reduces the levels of androgen in this particular disease. So also a very interesting approval. The approval of the so-called Vanza triple will be detailed later on by Brett in this presentation. Also, we have a wonderful story about this on Drug Hunter, and Brett will share all the details with us later. Moving on to the infectious disease space, we saw about 4 approvals in the infectious disease space. The first one is berdazimer. Berdazimer, kind of on the verge of being a small molecule, is basically a nitric oxide releasing agent here for a highly infectious skin condition. So interestingly, this can be applied topically. Then in terms of the other approvals in the space of infectious diseases, we pretty much saw a lot of beta-lactam antibiotics being approved. So here, this first combination with a beta-lactamase inhibitor, as well as a cell wall synthesis inhibitor, so a traditional beta-lactam, and this is being used for complicated UTIs. Another interesting molecule, also beta-lactamase, beta-lactam cell wall synthesis inhibitor, is this compound here. Also contains this other super interesting structural motifs that has an oxime next to an amide here, motive that you not that commonly see in drugs, and that is being used for Staph aureus bloodstream infections and specific skin infections, discovered by Roche earlier. Here's a very interesting strategy of this time not combining a beta-lactam with a beta-lactamase inhibitor, but with an OAT3 inhibitor. So that's a very interesting strategy that basically reduces the renal clearance. And it's a strategy that's kind of commonly used in the antibiotic field. Many antibiotics are actually cleared renally. So amoxicillin, for example, has 60 to 80% renal clearance. Cefampin, that we see here, has approximately 80% renal clearance. So dosing this with kind of an inhibitor of renal clearance, an OAT3 inhibitor, is something that's a really interesting strategy. Has also been applied beyond antibiotics, for example, with statins. Pravastatin, or also even fexofenadine has been co-dosed with an OAT inhibitor. Can also be used maybe preclinically if you want to look at renal clearance. That could be an interesting strategy for a preclinical model as well. Moving on to hepatology, that's an interesting area as well that has seen a lot of breakthroughs here as well. And Rory is going to detail the first approval for the indication of a MASH, which is resimetirom. So really interesting THR beta partial agonist. And Rory is going to share the discovery story and much more about this with us later today. We also had 2 super interesting compounds that are PPAR agonist approved in that space. They were both tried on MASH before, but ended up being approved for PBC, primary biliary cholangitis. The first compound here is a dual PPAR agonist. So just to talk a little bit about the disease PBC. PBC is kind of a chronic autoimmune liver disease that affects the bile ducts. So the small bile ducts in the liver kind of become inflamed and are being progressively destroyed in this particular disease. And that leads to an accumulation of bile in the liver that's resulting in inflammation and scarring of the liver. And PPAR, how is PPAR doing something against this? So PPAR is a group of nuclear receptor proteins that function as transcription factors. And in specifically the PPAR delta pathway, there is an enzyme that can be downregulated by PPAR agonism that's called CYP7A1. And basically, that's one of the mechanisms, that's one of the key enzymes in the biosynthesis of bile acid from cholesterol, and kind of downregulating bile in this disease is particularly helpful. But that's just one way PPAR is influencing it. The PPAR family also controls very complex cellular processes in lipid oxidation, metabolism, and fatty acid metabolism. So both of these compounds really interesting. Seladelpa, again, a more selective compound, more selective on PPAR delta compared to this dual agonist here. Interestingly, has been licensed by Gilead from CymaBay for about $4 billion. So very interesting acquisition there as well. In the space of dermatology, we have this interesting M3 receptor antagonist here for a skin condition, and it can be applied topically, which is very nice because you don't necessarily want to antagonize M3 systemically. M3 receptors are really expressed throughout different organs in the body, and controlling the antagonism with local application is something that's very interesting. Duroxolitinib is the deuterated version. Many of you will probably recognize this compound. It's the deuterated version of basically Incyte's first-in-class JAK inhibitor that was approved in 2011. So the deuteration, which was originally discovered here by Concert, who also were discovering the deuterated version of ivacaftor, and I guess Brett is going to talk about this a little bit later. So the deuteration here led to basically a reduction in oxidative metabolism, and therefore this compound is being used in alopecia areata. Last year we talked about Pfizer's JAK3 inhibitor relevance. And here with the, basically deuteration, you have a longer action on target, so you can have sustained inhibition of JAK with that particular compound. So very interesting kind of strategy to think about more generally. And deuterium is becoming more and more, common in approved drugs. In the respiratory space, we had a dual PDE3 and 4 inhibitor approved here. For a chronic obstructive pulmonary disease, COPD. And this is typically helping to prevent the exacerbation in COPD patients. This mechanism is also becoming increasingly more important, as we are seeing, for example, from Boehringer Ingelheim. They have a PDE4B inhibitor that just a few days ago they announced positive phase 3 results by meeting their primary endpoint. So it's a field, PDE inhibition is a field that we want to watch going forward. In the neurology space, we also had a breakthrough approval with Cobenfy, and Lou is going to cover the discovery story of that, so I won't steal his thunder as well. In the hematology field, we had 2 approvals. One is a HIF-PHD inhibitor for anemia and chronic kidney disease on dialysis-dependent patients. So, this is an interesting second-in-class compound here after the approval of daprodustat last year. So really an interesting field to be watched. One of the things that most of these HIF-PHD inhibitors carry is a black box warning due to risks for cardiovascular events, increased risk of cardiovascular events, and that prevented some of the molecules to actually be FDA approved as well. So it's an interesting new mechanism in the field of anemia, but also in higher doses, one has to be somewhat careful about this. Another interesting approval here was this complement factor D inhibitor in the space of PNH. So in PNH, which is actually an interesting disease, the basically complement system, which belongs to the innate immune system here, attacks the red blood cells, because in that disease, they don't have kind of an expression of a specific protein that prevents the complement system to attack them. The complement system typically attacks like bacteria, viruses, and foreign organisms. And, in this particular disease, the red blood cells are not expressing kind of the defensive factor. So a complement D inhibitor is alleviating that. In the cardiology space, we had 3 approvals. First one was this dual ETA and ETB receptor antagonist, and that's used in the treatment of hypertension. It was originally discovered by Actelion, but there was a demerger between Actelion and Idorsia. This compound was originally licensed then to Janssen, but Janssen licensed it back to Idorsia. Yeah, we also had an interesting approval here as a TTR stabilizer, acoramidis, which is a second-in-class, potentially best-in-class stabilizer coming after Pfizer's tafamidis. So this is a nearly complete stabilizer of TTR, which is a protein that's important in cardiomyopathy. It's a carrier protein that, for example, carries bound vitamin A or thyroid hormone, a transporter for the thyroid hormone. And basically, we have seen with this compound improved overall survival and reduced basically cardiovascular related hospitalization. So we have a really interesting story about this on Drug Hunter, showing how the binding of this compound is almost entirely enthalpy driven. And I would recommend everybody to kind of read up on this case study. It's also interesting that it had an academic origin here. Last in the space is lanidiolol, which is an interesting compound as well. And Ono originally brought this to the market. It was already approved in 2002 in Japan, and now also 2024, it made it as an FDA-approved drugs. We also have 3 diagnostics being approved. The first one is probably the most interesting one. So the first one is used in pre-operation for breast cancer patients. Basically, here upon the peptide cleavage by cathepsines and matrix metalloproteases, this becomes optically active. So basically, the doctors in an operation can see which cancerous tissue is, and which tissue is cancerous, and what they need to remove there. And then we had 2 other interesting diagnostic agents approved as well. And with that, I'll hand it over to Dennis to go and do a deep dive into the revumenib story. Dennis, stage is yours.
Thanks, Dennis. And to be fair, for the inventors of all of the menin-MLL protein-protein interaction inhibitors, a deep dive here of 2 slides is definitely not going to do the decades-long story here justice, but just thought we'd give more highlights into why these molecules are so cool. So revumenib is an oral first-in-class menin inhibitor for KMT2A-driven acute leukemias, including AML, some ALLs, and MPAL. The menin-MLL protein-protein interaction is a driver they're driver mutations in a number of these different leukemias, where the MLL forms these fusion proteins with all kinds of fusion proteins, similar to the idea behind BCR-ABL. The ABL BCR fusion protein uniquely drives certain leukemias. And this target, the menin-MLL protein-protein interaction, has been really attractive because it's not essential in normal proliferating hematopoietic cells, but is driving these pathogenic cells. This is a long history going back to the University of Michigan, and we'll see a little bit more of the chemistry on the next slide. Sorry, we can stay on this slide for now. Clinically, it's really a game changer because, as many folks know, AML has been incredibly difficult to treat. When it manifests, it's often very aggressive, and there are very few options for it. In the last 2 decades, there have been almost no new therapies approved except for targeted therapies like FLT3 inhibitors, which work for a subset of patients. And the menin-MLL mutations drive quite a large double-digit percentage of these leukemias. So it's a rare new mechanism, highly effective, and it was so in the treatment of leukemias. One of the side effects that you sometimes see when drugs work is differentiation syndrome. And so what happens in some of these cancers is the leukemic cells can basically, so leukemia is a cancer of blood cells. When blood cells divide, they usually start with hematopoietic stem cells that progress into differentiated cells. So you have, these stem cells, and they turn into these blast cells. Those blast cells give rise to, further progenitor cells, and then ultimately those become, things like platelets or red blood cells, white blood cells, etc. What can happen in these leukemias is these cells get trapped in one of these dividing stages, those dividing cells basically never differentiate, and they just overwhelm the blood. And in some of these patients, right, the blood it's so overwhelmed with leukemic cells that the blood becomes visibly thickened by the sheer amount of tumor cells in the blood. So a side effect that can happen is some of the drugs that work for leukemias promote the differentiation of these leukemic cells into the downstream, into differentiated cells which die. And when they do this, they release cytokines. And if you basically are treating these leukemias almost too quickly, that the cytokine release can be overwhelming and lead to this differentiation syndrome, which can be lethal. But it is a sign of the drugs working. So early in the story of these molecules, clinically, I think, I can't remember if it was Syndax or the other company, Kura Oncology. I think both experienced this. Early on, they had a FDA clinical hold for these programs because of the differentiation syndrome. And so, stocks of both companies tanked. I think there was some concern about this area in general. Some people left the menin MLL space, but I think a lot of people watching the space were like, that's a clear sign that this is working. This clinical hold is likely going to be resolved. These are probably, this is a very clear sign of efficacy. Why are people so afraid of this area? And sure enough, the hold was resolved, and now there is an approved drug, and there is likely a second approved drug in this area coming. So an interesting case where an FDA clinical hold does not necessarily mean that things are going wrong. In fact, it may be a sign that a drug is really working. All right, so on the next slide, I think these molecules are all beautiful protein-protein interaction inhibitors, and there's a long history going back to some elegant work done at the University of Michigan, a collaboration between two groups, structural biology group and some folks doing some more chemistry. The starting point, interestingly, for two of these drugs that are both either approved or, knock on wood, about to be approved, are these fragments that were discovered through structure-based drug design. So if Dan's still in the audience, I'm sure this is something that he'll very much appreciate. So you have this pyrimidine on the left, which is over micromolar in Ki. Interestingly, the two different groups took this starting point in different directions and ended up with very different looking molecules. But both ended up with very potent chemical matter, as you can see, as this compound progressed, with that spirocyclic core as a piperazine isoster, which is pretty cool. They ultimately got to picomolar binders, like that compound on the right, which is not this drug, but is a closely related analog. One of the cool things when you look at the crystal structures of these things, and if you look at the full article, there's a lot more detail on the chemistry here. But one of the cool things is if you look at that middle part of the molecule that's highlighted in that black box, you can see that basic amine sort of is sandwiched between two pi clouds of two tyrosines. And so it's a sort of in a pi clamp. And it also makes this interesting water-mediated hydrogen bonding interaction as well, which is pretty cool. So that amine core, you'll see, is pretty important in all of the menin MLL targeting molecules. These molecules all sit in this hydrophobic pocket that helps block the menin MLL protein-protein interaction. It's interesting that they do bind to the MLL. They both bind to menin. And on the next slide here, if we look at all the molecules that have emerged, you can see, there's typically that heteroaromatic left-hand side, which, sort of related to that starting fragment. You typically have this amine or amine-related group in the middle. They're highlighted in green. And there have been quite a few active players in this space. So Syndax is the first to achieve approval. Kura Oncology, I believe, licensed the University of Michigan chemical matter and has a registrational trial right now ongoing in phase 2 with ziftomenib, likely, knock on wood, likely maybe a second approved option shortly. And Bioimmunofusion, interestingly, has a covalent version that they are using in AML. And this was actually in the news quite a bit, not for AML, but actually for their reports that these things, that this may be impactful in diabetes, which is quite a surprising different area for this target. Daiichi Sankyo, Johnson & Johnson, Sumitomo have all been active in the space. As you can see, the chemical matter is related, but hopefully these will provide some very exciting options for the AML patients that do have these mutations, AML, ALL, or MPAL patients. So I highly encourage people to dig into these case studies. There's a lot of rich drug discovery, biology, and chemistry here, but definitely beautiful molecules to pay attention to.
All right. So I think we've talked today about how long-running some of these drug discovery stories can be, and I think I'm going to have the record today. This is a drug discovery and development story that's about 130 years in the making. So I'll be talking today about Cobenfy, formerly known as KarXT. This is an oral muscarinic acetylcholine receptor modulator combination therapy that was approved in 2024 for schizophrenia. It's also currently in phase 2 and phase 3 trials for Alzheimer's disease. So this combination therapy was discovered and developed by Karuna Therapeutics, which I think folks know was acquired by Bristol Myers Squibb in 2023. And this story is really exciting because this is the first new FDA-approved schizophrenia treatment with a new mechanism of action. So this hasn't happened really in decades. And this molecule seems to have effects on both the positive and negative symptoms of schizophrenia. And as folks know, therapies for schizophrenia generally address the positive symptoms, but not so much the negative symptoms. And so this story, like I said, has been going on for a long time. And it's really interesting that one of the molecules, xanomeline, was actually first synthesized in the 1990s in a collaboration between Eli Lilly and Novo Nordisk. The other part of the combination therapy, tropium chloride, was actually first synthesized in the 1960s in Germany by Robert Fleger. And then this whole combination strategy that we'll be talking about today actually was first pursued in the 1950s at Emory University. And all of this work on these muscarinic acetylcholine receptor agonists really stemmed from a natural product, ericoline, that was first isolated in the 1880s by a German chemist, E. Jans. So really this story's been going on a long time, and this is this nice combination of a very old story with a very sort of new outcome. So what is xanomeline? Xanomeline is the CNS penetrant M1, M4 selective MCHR agonist of the combination therapy. And what was found is that it binds to both an orthosteric site and an allosteric site in the M4 receptor. And then tropium chloride is a peripherally restricted, unselective MACHR antagonist. And so that's in the combination therapy, really to try to reduce the GI side effects of xanomeline that include nausea and vomiting. So why is this important? This is important because schizophrenia is a disease that afflicts a large number of people worldwide, about 24 million people worldwide. And as most folks probably know at this stage, most antipsychotics are dopamine D2 receptor modulators of one type or another, often with polypharmacology. And so there are treatments out there for schizophrenia, largely addressing the positive symptoms, but 30% of patients are actually treatment resistant to the current battery of therapies. And as I've mentioned before, these therapies generally address the positive symptoms like hallucinations or hostility or delusions, but they have little benefit for the negative symptoms like blunted affect and emotional and social withdrawal, cognitive impairment, such as deficits in attention, memory, and executive function. Additionally, like I said, there are a lot of side effects with the current therapeutics that are out there, including sedation, extrapyramidal motor symptoms, weight gain, as well as other symptoms that further complicate patient compliance. And indeed, 42% of patients actually stop treatment within 18 months of starting therapy. So clearly, I think this shows that there is still an unmet need out there for new treatments that address all of these symptoms and don't have the sort of debilitating side effects. So if we move to slide 2, you can see that this slide really illustrates the challenges and the opportunities of pursuing MACHRs for schizophrenia. So these receptors are expressed ubiquitously throughout the central nervous system and the peripheral nervous system, and have a lot of effects on the parasympathetic nervous system, as you can see here in this figure. So one of the challenges is trying to bind to and modulate the activity of the muscarinic receptors you want to target in the tissues you want to target without hitting them in tissues you don't want to target. So that's the genius of this combination therapy approach, where xanomeline is an M1, M4 selective agonist, gives you the effects you want in the central nervous system, and tropium chloride is a pan muscarinic antagonist, basically ablates all of the side effects of this therapy. And like I said before, I see at the bottom of this slide that this therapeutic hypothesis was first pursued in 1957 at Emory University. So really a long-standing sort of effort. So if you move to the next slide, xanomeline has been found recently to actually have very interesting pharmacology, and that there was actually an X-ray crystal structure first solved in 2023 that I think surprised a lot of folks when they first saw it. And that's because xanomeline was actually found to bind to the orthosteric site of the M4 receptor, but is also an allosteric binding site in the sort of extracellular domain. This is where other allosteric modulators like emraclidine bind. So really, a very simple concept that's been around for a long time, a lot of molecules that have been around time, but it basically took a group of people who were dedicated to this therapeutic hypothesis and kept pushing on it for a number of years. And here we have today a new mechanism of action for treating schizophrenia, and hopefully a way that benefits a greater proportion of patients. So really an exciting story. And what I've shown here on these 3 sides is just a sliver of the story that you can find on Drug Hunter. So if you want to look at the article, go to Drug Hunter. You can see all the work that's been done by a lot of other companies historically. And then you can also see a number of other companies that are working in this space, seeking to try to take it to the next level. So I think this is one of the most exciting approvals of 2024, and I'd love to take any questions after we're done with our presentations. Thanks.
Hello, everyone. My name is Brett Buchser. I'm going to talk about a leaf trek today. In full disclosure, I worked for Vertex several years ago, so I'm a little bit familiar with what was going on through the time period of the discovery of these molecules, but everything that's in this article that I wrote for Drug Hunter is from publicly disclosed information. So a couple highlights just right off the bat with these interesting molecules. You can see that we have a macrocycle on the far left, vanzacaftor, with 3 curiously spirocyclic cyclopropyl rings down at the bottom. Very neat, interesting molecule. And then I'll skip tezacaftor in the middle, talk about that a little bit later. And then deutivacaftor on the far right has these 9 deuteriums, which help make this compound more metabolically stable and improve the half-life of it. So a couple highlights that are in the full article. There is a concept of chameleonicity that I talk about with the discovery of deutivacaftor and its predecessor, ivacaftor, as applied to a rule of 5 molecule. This is a concept that's typically used to describe how beyond rule of 5 molecule can change its shape in order to assume a more lipophilic form and have better permeation through barriers in the body and cells. And so in this case, this may be the first application of the concept to a small molecule, like rule of 5 molecule, like ivacaftor, deutivacaftor. These molecules, they also change their shape somewhat in order to become less polar. And I would argue that that feature really helped this molecule move forward and become an approved drug because of its changes on the potency property profile. I talked about the T-butyl group being per-deuterated a second ago. Also in the article is the application of a structure-based hypothesis that Vertex employed at some point, and they published this. And what it says is that compound length can decrease PXR activation, which impacts CYP3A4 induction. And this was an important feature, which I'm going to talk about when I talk about all the molecules together. The key compound length, if you want to remember it, is the number 14. Just remember the number just one after 13, 14 angstroms. If your molecule is beyond 14 angstroms, you might have a good possibility that it won't be an inducer of CYP3A4. The macrocycle improves a non-macrocycle, elexacaftor, in potency, in the dosing potency. You only need to dose 1/5 the quantity of compound compared to elexacaftor. So the macrocycle probably definitely played a role in that. So now we'll go to the next slide, and I'll talk a little bit about the mechanism. So here's a cartoon which explains a little bit about how cystic fibrosis happens because of cystic fibrosis transmembrane receptor mutations. So the CFTR protein is this little dark blue element at the top of and so this protein is responsible for the chloride and other anion flux through the membrane, and there are approximately probably more than 2,000 mutations that are genetically inherited that cause cystic fibrosis. And so these mutations have been classified, and the classes are listed on the right. So class 1, 5, and 7 mutations are DNA RNA errors, which either allow no CFTR to be synthesized, or the CFTR that's made is non-functional. Class 2 error mutation is a protein misfolding, which when the protein doesn't fold correctly, it just gets marked for degradation and it gets put through the proteasome and it's degraded and it's non-functional. So the class 2 mutation was targeted by Vertex and other companies with correctors, and correctors are molecules which are designed to allow the protein to correctly fold so that it can traffic to the cell surface. Class 3 and class 4 mutations impact the ion channel gating and the conductance of the protein. And so class 3 and class 4 mutations are addressed with the potentiator approach. And then the class 6 is an accelerated turnover from the cell surface. So the approaches that were used by Vertex to discover drugs were the class 2 and class 3, class 4 mutations. So go on to the next. So these are the 6 molecules that are part of the molecular journey to the newest drug, Leaf-Trek. So I'll start with ivacaftor in the top left, the potentiator. So when Vertex started this work in around the year 2000, the work was already initiated by another company called Aurora Biosciences in San Diego. Aurora Biosciences was a company which developed fluorescence assays, and they wanted to apply these fluorescence assays to ion channels that impact the disease. And one of the ion channels was CFTR, the other for CF, and the other is the NAV ion channel, which impacts pain. And Vertex bought the company in year 2000 and progressed both of those projects to develop drugs. The NAB one, of course, we just heard about a couple weeks ago, is the suzetrigine drug for pain that they got approved. So this is the other one, which is based on CFTR. So there are two or three important cellular assays that are used to identify compounds like ivacaftor and lumacaftor. So for ivacaftor, there are actually three assays. The first assay is a high throughput assay based on a recombinant cell derived from mouse that has a F508 deletion, and the F508 deletion impacts the folding. But if they run the assay over a long period of time, they get enough CFTR to express at the surface that they can see an effect on chloride ion flux. So this is the high throughput assay. And then the next assay is a more selective assay, which is derived from human bronchial epithelial cells from actual donors that have the genetic defect, the mutation in their own CFTR. And so human donors donated these HBE cells, and assays in that help confirm that in a human, you can have this potential impact on ion flux. For ivacaftor specifically, there was a third assay, which is the F508 del G551D mutation. It's a combo mutation too on the two alleles. The G551D importance is that that's a mutation which specifically impacts the gating and conductance of CFTR. So this is specific for, if it works in this, the Vertex found that in this assay, when they eventually got to ivacaftor, that the performance of the ion flux, the chloride ion flux, was equal to half of what a normal person had. And so they felt confident that if they went into the clinic with just those patients that had this mutation, they would have a good chance of success, and they did. And they got their first approval in 2012. So in parallel with the discovery of ivacaftor, the potentiator, they're also trying to find correctors. And so they, in parallel, they're assaying compounds, which eventually led to lumacaftor. They had the first assay I mentioned, they ran more than 160,000 compounds through it, and they eventually came up with a hit, which the only similarity that hit has to lumacaftor is that it has an amide in the center, and the left and right sides of it are actually different, and they were both optimized to get to this molecule lumacaftor. And I should note that there are MedChem publications which talk about the full story of ivacaftor and lumacaftor, and we cover it in the article that's highlighted at the bottom. So for lumacaftor, this molecule performed in combination with ivacaftor in assays better than lumacaftor alone or better than ivacaftor alone. And so they progressed this combination of ivacaftor, lumacaftor into the clinic. And what they didn't know, and they didn't know from preclinical assays, was that lumacaftor is actually a molecule that causes induction of CYP3A4. And because it causes induction, ivacaftor gets degraded faster than it does normally. You have a drug-drug interaction. So you have to actually double the dose of ivacaftor compared to ivacaftor alone in the first approval. So this is a problem, and because this is first in class, it's the first time that a corrector has been approved in combination with a potentiator, the first time this type of approach has ever been taken, and FDA approved it, and it helped a lot of patients when it was first approved. But in parallel, once they noticed that issue in the clinic, Vertex began to was always looking for better correctors within this class. And they eventually ended up with tezacaftor, which is not an inducer. And that molecule combined with ivacaftor won approval in 2018. In these corrector discoveries, they noticed that there was a second structure type that also had a correction effect. And they explored the optimization of that second structure type and ended up with the molecule lexicaftor in the bottom left corner. So now you have a molecule, and it was shown that this molecule actually has an additive effect upon tezacaftor and ivacaftor in the clinic. So the three molecules together, two correctors, one potentiator, became the first triple combination, Trikafta, which is effective for 90% of patients. So amazing, and this was in 2019. So even with that nice achievement, Vertex is still optimizing, and they're pushing vanzacaftor and deutivacaftor through the clinic with vanzacaftor and deutivacaftor with tezacaftor as a combination. And the advantage is that vanzacaftor is dosed at 1/5 of the dose of elexacaftor. Deutivacaftor with these deuteriums increases the half-life. So now, in all the other previous combinations, you had to double the dose. You had to take ivacaftor twice a day, but the correctors you only had to take once a day. Now with deutivacaftor, you can take the whole triple at one time, and because you can take deutivacaftor, only required to take once a day now. So there's a pretty picture which shows how these three molecules interact with the CFTR in this slide. And so this is from a Science 2022 publication, which shows ivacaftor, tezacaftor, and elexacaftor all cooperating to correct the CFTR protein that is missing F508. And this paper has a lot of details about what these molecules specifically do to correct the channel. One thing they do point out is that while ivacaftor impacts the channel performance as a potentiator, and tezacaftor impacts the folding of the protein, elexacaftor actually does both. Elexacaftor actually helps with folding, and it impacts one of the nucleotide binding domain gating of the channel. So it has this double effect as a corrector. So the final tie-in to the leaf track is in the next slide. So vanzacaftor is a macrocyclic form basically of elexacaftor. And so this is a representation of elexacaftor in that cryo-EM structure, and vanzacaftor just from a PubChem 3D structure. And you can see how vanzacaftor in its natural state perfectly overlays almost with the elexacaftor bound state. And so you can see here visually how vanzacaftor assumed the proper conformation of this type of corrector and helped it to achieve a higher level of potency compared to elexacaftor. So thank you very much. That's all I'm going to talk about here. If you want to read more, there's a lot of more stuff about the story that you can read in the Drug Hunter paper. Also, I would say I'll point you to my LinkedIn post from last week where I talk about the chameleonicity aspect in more detail. Thank you.
Yeah, thank you, Brett. So yeah, I'm going to cover inavolisib, which is Genentech's selective, super interesting mutant selective PI3K alpha monovalent inhibitor and degrader. And I think this story's like really it spoke to me because it combines kind of three things. It's got this really interesting chemical structure. It's got this cool 6,7,5 tricyclic core with this really interesting difluoromethyl oxazolidone kind of bottom piece. So it's got that interest, it's got that chemical interest. I think it has a really interesting kind of development story and a mode of action, which kind of really speaks to the Genentech team's kind of appreciation of detail and kind of really digging into their data as how they're able to get to this kind of novel mode of action in a kind of area that has been hotly pursued by a number of different companies. And really, what matters most about drugs are really interesting outcomes in the clinic. In breast cancer, which has been heavily pursued again over the last decades, but in this HR positive, HER2 negative, which was a breast cancer which is approved for managed to have really some outstanding kind of results on top of what's already been done there. So as I said, it's a PI3K alpha isoform selective inhibitor. And this is kind of key because a lot of the inhibitors which have been kind of in the clinic and which have been approved actually have side effects which are due to kind of off-target, mostly due to GI effects, which are caused by PI3K delta inhibition, and also due to wild-type inhibition, because actually there's this feedback loop with the RTK signaling pathway, where inhibition ends up kind of paradoxically increasing the effect of the signaling here. Also, this tends to lead to this wild-type inhibition tends to lead to hyperglycemia, which has been really kind of the limiting factor in using this clinically because of these high rates we see of grade 4, grade 3, or grade 4 hyperglycemia in the clinic. So inavolisib works but through this high alpha selectivity, and also it has these two interesting mechanistic aspects. One, in that it's where you have high RTK signaling, it's actually more effective as an inhibitor. And actually the fact that it is a degrader selectively of mutant forms of this, the P110 alpha subunit, mutant forms of this are degraded while sparing the wild type. And this high selectivity is key not only for kind of these off-target effects, but really the key to kind of advancing breast cancer treatment in the last years has been these combination therapies. And so by having a really clean off-target effect profile, you are able to dose with these other drugs. So inavolisib was approved in combination with a estrogen receptor blocker and palbociclib, which is a CDK4, CDK6 inhibitor, which is known to have kind of toxicity. So by having a clean partner, they're able to kind of really get high dosing and be able to have a big effect. So if you skip to the next slide. So the discovery story of inavolisib at Genentech came from when they were looking at their previous preclinical candidate, tazalisib. And they noticed that actually this compound had showed stronger anti-proliferation effects versus the other PI3K inhibitors, and more so than kind of like the alpha selectivity, the isoform selectivity, or the cellular potency would suspect that it should do. And digging into this was how they found this degrader effect that was being used by tazalisib. So kind of breaking this down into a smaller molecule, looking at compound 1 here. This was a great starting point because this showed the isoform selectivity they were looking for in terms of alpha over delta, but it didn't recreate that P110 alpha mutant selectivity here. Changing the bottom piece, this triazole that they used in the compound 1, they looked to move to a kind of non-aromatic kind of cyclic core here, cyclic headpiece here, which would hopefully give them better properties, better PK profile, but also have these hydrogen bond donors, which were key to the kind of complex kind of water engaging hydrogen bond networks, which were key to the selectivity here. This actually maintained and improved the selectivity they saw of alpha over delta, and actually now start to have really nice bioavailability and kind of good clearance numbers. Changing the top piece here from this O-linked to this N-linked was also important now to start getting this degradation that they were seeing. And the mechanism which they kind of invoke for this is that this P110 alpha subunit is inherently less stable. And actually it ends up getting localized to the membrane, and there it's degraded through the E3 kind of ligase ubiquitin proteasome system. And actually by having this, they were getting improved selectivity for that. When they finally got to compound 32 or inavolisib, it was by kind of switching to this alanine up there. And this now shows like really nice PK profiles, really nice activity, and this really strong selectivity now over 300-fold for alpha over delta, which was really able to allow Genentech to push into the clinic and to really dose up. And so in the clinic, it was approved based on its 57% reduction in the risk of disease progression in combination with fulvestrant estrogen receptor blocker and palbociclib, the CDK4/6. So really doubling the progression-free survival which the patients were seeing. So they've got multiple other phase 3 trials that are ongoing to try and expand this indication. But I think this is a really kind of great story. And like I said, I think the chemical matter here is really interesting. I think at heart, I'm still an organic chemist. So if we go to the next slide, we can kind of go through some of the medicinal chemistry synthesis of this. And classic chiral pool starting material. So actually takes me back to grad school where I did this first reaction on 100-gram scales, and it's a really useful and flexible building block here. And from that, they're able to include to bring in this difluoromethyl piece. This is actually really interesting in the crystal structure. You see a non-canonical hydrogen bond between the fluorine and a serine in the pocket, which is like a nice part of how they get selectivity actually for the for alpha over delta. You can break open this acetal and then selectively incorporate a nitrogen in the cyclized background. And this is the kind of thing that I think everyone should have, this super interesting looking building block in that maybe their building blocks draw in the lab. This would be the kind of thing that you'd want to tack onto a bunch of different analogs to see what it can do. If you go to the next slide, the way the team kind of brought together this core, it's kind of got this neat double cyclization, so you're able to form the 7-membered ring, and this actually opens up the handle that you can then form this imidazole, the second cyclization. They're actually not able to do a selective iodination to bring in the handle, which they need to bring in that oxazolidinone piece. But what they can do is they can do a double iodination and then selectively burn off the unwanted one for a Grignard exchange and then quenching. It's going around the houses a little bit, but it really works in terms of selectivity to bring in that iodine. And then you've got two selective couplings, Ullmann-Goldberg couplings, to bring in the oxazolidinone piece and then the alanine piece, which then can be finally converted to the primary amide. And yeah, like I said, I think this is a great story, and I encourage everyone to go onto this case study and read it, of this combination of kind of great chemical matter, great kind of scientific attention to detail to bring out that novel mechanism, and then great effects in the clinic. So yeah.
All right, thanks so much, Matt, and thanks again, Dennis, for organizing this. This is really awesome, and it's been a lot of fun. So today I just wanted to present the landmark approval of resimetirom, or the brand name is Rezdiffra, the first and only FDA-approved treatment for NASH, increasingly referred to as MASH, and which really marks a pivotal moment in liver-targeted small molecule drug discovery. So just a little bit about some background that's here on the slide. So this molecule was granted FDA accelerated approval almost a year ago now, March 2024, for the treatment of MASH. And it's also this first-in-class THR beta agonist and the first ever approved compound to treat this indication, as Dennis alluded to earlier. So just some background about MASH and NASH. It affects an estimated 3 to 5% of the global population. So if you think about that, that's a huge number. And it's also one of the leading causes of liver transplants. And really, yet developing drugs for NASH has been extremely and extraordinarily difficult. And I'm going to return to that concept, why it's so difficult here, I think, on the next slide. So non-alcoholic steatohepatitis, as I said, now increasingly referred to as metabolic dysfunction. Associated steatohepatitis is this progressive liver disease that stems from non-alcoholic fatty liver disease. And it's often characterized by this excessive liver fat accumulation, chronic inflammation, and as well as progressive fibrosis, which can ultimately lead to cirrhosis, liver failure, and even hepatocellular carcinoma. So also on this slide, we have a bit about the target rationale. So resmetirom is this selective thyroid hormone receptor beta agonist, and it's really designed to improve the liver lipid metabolism and prevent fibrosis progression. So it turns out that the thyroid hormone signaling plays a key role in lipid homeostasis. And by selectively targeting THR beta in the liver, resmetirom helps to reduce hepatic fat accumulation through fat oxidation, lipid oxidation. It also decreases inflammation and ultimately prevents the progression of fibrosis, which is super notable here. So if we go to the next slide, so it's originally discovered at Roche Nutley, and resmetirom was identified as compound 53 in the original patents that were emerging early on in about 2006. And as you can kind of see based on the chemical structure here, it's a very close analog to triiodothyronine, also referred to as T3, which is an essential thyroid hormone. And so that's ultimately the starting point for resimetirom. Something I just wanted to point out here are some structural similarities. So as you can see on the resimetirom, there's this pyridazonone group, which kind of mimics this phenol group on T3, as well as there's this acidic cyanoaza uracil motif on the top, which mimics the carboxylic acid group. And really, it's this group that helps to drive the selectivity for resimetirom between THR beta and THR alpha. So, early on, the Roche team utilized structure-based drug design with the T3 bound to THR beta, and that helped guide their medicinal chemistry campaign. But ultimately, Roche outlicensed the program to Via Pharma in 2008. And then after the Nutley site closure in New Jersey. And then later, this asset was acquired by Madrigal in 2011. So, ultimately, it was then 15 years from this acquisition from Via to then Madrigal, really led by the founder and the CMO of the company, Becky Taub, and who ultimately was a huge champion for this molecule and helped to bring it to the market. So I think this is really one of these really nice, important lessons of scientific champions. Them recognizing promising molecules, keeping them alive, even after major pharma companies site closures and things like that. Just some notable properties about the molecule would be that I've kind of alluded to this, that resimetirom is 28-fold more selective for THR beta over THR alpha, which is really important because THR alpha is highly expressed in cardiac and bone tissues, and so avoiding THR alpha is super important for a clean tox profile. It also has this really amazing liver targeting properties. So, if you look at the distribution in mouse studies, it has about an 8-to-1 liver to plasma ratio, which is super important for reducing systemic thyroid hormone effects. It also has a very low dosing. So, once daily oral dosing of less than 100 mg QD helps to mitigate drug-induced liver injury. And, oftentimes these patients are on multiple drugs. And so having a low QD dose is super important with mitigating tox. So if we go to the next slide, so why has it been so difficult to develop drugs for NASH? And so oftentimes people will characterize this disease as being a very slow progressive disease. It's a chronic disease, oftentimes requiring very long trial durations. It often has there's up until now, there's been a lack of clear surrogate biomarkers for clinical benefit, as well as this reliance on sequential invasive liver biopsies. And so patients, tend to, maybe not comply as well when they have to go in for liver biopsies routinely to evaluate the progress on the drug. It also just has a high safety bar. So NASH drugs must be safe for chronic use, as I kind of alluded to. Oftentimes these patients are on various other medications as well. So, despite immense investment in the NASH drug development, this area has been plagued with very high profile failures. And one can think about the Intercept's FXR agonist, obeticholic acid, which, kind of serves as a cautionary tale, right? It was able to show promising fibrosis reductions in the clinic, but was ultimately then rejected by the FDA due to safety concerns, including proctitis and other liver injury issues. And there's other, as you can kind of see on the slide here, there's other high profile failures, including Gilead's ASK1 inhibitor, which failed in phase 3 due to lack of efficacy. Canadis's pan-caspase inhibitors failed to show meaningful clinical benefit. And then I think we all remember Genfi and Ipsen's dual PPAR alpha delta dual agonist, which failed to meet its primary endpoint. Ultimately, we know that this was approved also last year, as Dennis alluded to during his presentation for PBC. So if we just go to then the last slide, here, it is just some of the phase 3 clinical data. Ultimately, resimetirom was approved via an accelerated approval process. Some of the data is listed here. I don't need to go through all of it, but ultimately the data shows that resimetirom led to a reduction in NASH, no worsening of fibrosis. There was also fibrosis improvements by at least 1 or greater stages of fibrosis. Fibrosis is a spectrum, and so there's F0 up to F4. And so 25 to 25% of those patients were observed to have a fibrosis improvement. The drug also led to LDL cholesterol reduction, which is great. And so, ultimately, the next big question is whether resmetirom's surrogate endpoint approval will translate then into long-term clinical benefits and specifically preventing cirrhosis and improving survival. And then maybe a last question here that we're all waiting for is, will the long-term studies confirm a reduction in liver-related mortality to receive this full FDA approval? So also on the slide here, I just have some other THR beta agonists that I kind of wanted to call out. So I think the closest competition for Madrigal right now is Viking's VK2809, which is a THR beta prodrug. This is currently in phase 2b, and this drug right now, has some very promising phase 2 data. There's also this other notable earlier THR beta agonist from Caro Bio, which was actually discontinued to excuse me, cartilage toxicity in canines. So that was another high-profile failure, early on in this space. I think that was back in 2011 or so. And then I just wanted to end on, there's other drug classes now to watch that we're kind of keeping a close eye on for NASH and MASH. Dennis alluded to this as well, this PPR delta agonist seladelpar, which was developed by CymaBay, now acquired by Gilead. We know that it was approved for PBC last year, but it also has shown anti-inflammatory, anti-fibrotic effects in liver disease. And interestingly, this molecule was investigated early on for NASH, but it was ultimately discontinued because of atypical histological findings. And CymaBay decided to terminate the clinical studies for this molecule for NASH. But interestingly, an interesting turn of events here was that an independent panel review determined that there was this, the similar presence of atypical liver histological findings both before and after seladelpar treatment. So, they came to this conclusion that these histological findings were not because of the drug. And so it's going to be interesting to see if Gilead or any other company picks up these, pursues these PPAR delta, for this indication. There's lots of other targets, FGF21. Obviously, GLP-1s are going to be very interesting to closely follow to see how they address the obesity side driven form of MASH. And so we're keeping a close eye on that. And, like I said, there's many more. So this is a very exciting area to watch in the next couple of years ahead. And, at this point, we can open the floor up for some questions.
Yeah, first let me thank everybody for presenting, and want to let the audience know all of the case studies that you saw presented today are available on Drug Hunter in much more detail. So if you're interested, if that piqued your interest, which we hope we could, please go to the website and check out these case studies, and you'll find much more details. I also want to take this opportunity to thank our entire team. You saw a big part of our team, but not the entire team. So I want to call out, a few new hires that we made. So probably some of you know John Ovington joined us last Monday. We're very excited for him to join as head of data. And we also are joined by Chase Smith, who has also more than 30 years of experience in the medicinal chemistry field, both in academia as well as in industry. And again, huge things are going on behind the scenes. And we're the fortunate ones who get to present these case studies here to you. But really, this is a teamwork, a dramatic effort by everybody on the team. And we want to just take this opportunity to thank everybody who's working very hard behind the scenes to make Drug Hunter possible. And with that, again, thank you to everybody. And now I'm going to open it up to questions from the audience. So let me quickly unshare my screen, and I'll double check if I find questions in the Q&A box. Yeah, and, we have 5 questions already in the Q&A box. That's great. So let's go with the first one. Looking at those antibacterials, what is the industry not doing? Why is the industry not doing much more drug discovery? Perhaps a topic for a future drug hunter meeting. Yes, that's definitely something we can look at. And yeah, John has also been working in the infectious disease space, as have Matt and I. There are definitely a few very interesting case studies on Drug Hunter available that I would definitely recommend to check out. Another question is, I'm wondering how the team addressed the issue of the Michael acceptor in elafibranor. So yeah, maybe I can take that one. Rory, do you want to say anything about this?
I guess the only thing, so I haven't done a deep dive into this. I saw this question come in. I haven't done a deep dive into this particular molecule. But
Yeah.
clearly what we're seeing is, these covalent handles are becoming more prevalent in drugs, and so it doesn't seem that companies are as afraid to incorporate these motifs as perhaps they once were. So
Yeah, I can comment on this really quick. Actually, I'm very familiar with the elafibranor story, so that's actually a very interesting question. As people in the audience may know, the reduction of this double bond in the enone actually is the active metabolite in elafibranor, and also drives the clinical effects of elafibranor. It's also a dual PPAR alpha delta agonist. So actually, it turns out that if you look at the PK data comparing elafibranor to that metabolite, that the exposures of the metabolite are 5-fold higher. So essentially, people are thinking that the metabolite is driving most of the pharmacology action. Now, the question that I had that I don't think is answered yet, so why did they not just go with the reduced version of it, with the ketone as opposed to the enone? I will do some speculation here. Potentially, this is due to a better PK of the enone as compared to the ketone. Ketones, tend to be somewhat unstable, forming hydrates and whatnot. So potentially, they decided to go with the enone as opposed to the ketone. But again, that's somewhat speculation. But that's a little bit more detail on elafibranor here. Let's see what else we got. Oh, a question about xanomeline. Maybe Louis, you can take this. Do you have any insights on why, despite the long-standing awareness of the combination idea, the molecule was not pursued in clinical studies until 2020? Oh, sorry.
You're muted.
You're on mute. Sorry. Yeah.
Sorry, thank you. Every approved drug has at least one champion. Usually takes a team of champions. There's at least one champion in discovery, at least one champion in development, at least one champion to get it approved, at least one champion to get it successfully commercialized. So in this case, the champion clearly was, Stephen Paul of Eli Lilly. And I'm sure he had a very difficult time convincing leadership to advance the xanomeline project, given all of the work that had been done in the muscarinic receptors from the 50s to the 80s to the 90s. And, we probably didn't do enough justice to the complexity of the pharmacology. You have 5 muscarinic receptors. They're expressed all over the body. They have all sorts of effects, both positive and negative, depending on if you're activating or inactivating, where it's at, and so forth. And so with all of that effort that was put into this family of receptors, and nobody had really succeeded in getting an agonist or an activator across the line, it was probably pretty tough. So there was positive results that were seen in Alzheimer's trials for xanomeline in the 1990s. There was also a clinical trial. I think it involved 20 patients. It was a collaboration between Indiana University and Eli Lilly, the results of which were reported in 2007, where again, they saw some positive results in in schizophrenic patients, but there were side effects. It's probably no surprise. I think that was actually reported in 2008. It's probably no surprise. Stephen Paul formed Karuna in 2009. And as we understand it from looking into this area, apparently xanomeline was paired with dozens and dozens of different pan-muscarinic receptor antagonists, and they probably had to find the one that worked the best with at the right dose and so forth. And that wound up being tropium chloride. And so, I believe they initiated their clinical trial with that particular combination, their first clinical trial in 2016. So, with decades of effort and decades of failure, it really probably took a tremendous amount of dedication. I would love to have Steve Paul in for a flash talk to tell the story, the backstory of this Herculean effort. But I don't know for sure, but if I had to guess, it was somebody with the dedication and the resources to get this drug all the way to the goal.
Yeah, thank you, Lou. Maybe a question for Dennis about the earlier differentiation syndrome that you talked about. Somebody in the chat is asking, is the differentiation syndrome similar to a cytokine storm?
I think effect-wise, yes, in terms of it's the cytokine release that drives the the poorer effects that the patients experience, but different in terms of cause. I think with cytokine storm is typically in a reaction to, something other than the cells differentiating. But yeah, similar kind of idea.
Yeah, thank you for elucidating that a little bit more. And then question for Matt here on inavolisib. 2 questions here. Any idea about the steady state turnover of PI3K wild type or mutant, and whether this is what the drugs potentiate? And another question would be about the recommended dose, and what thoughts you have on the toxicity of inavolisib.
Yeah, so with regards to the first one, there's a really nice cancer discovery article that really goes into the mechanism of this. And they really do think it's about potentiating the kind of natural feedback mechanism rather than there's actually recruiting the P110 alpha as a neo-substrate to a new E3 ligase. The wild type protein is just more stable. So I think I've got the numbers here. It's got a half-life of 27 hours, whereas one of the mutants, H1047R, actually has a reduced half-life of about 10 hours in the basal state. And then when you dose with inavolisib, this comes down to 4 hours. And I think actually one of the key mechanisms by how this works is that inavolisib induces a conformational change that then leads to membrane localization. So you actually get a speeding up of the membrane localization, which is where the UPS system, where the E3 ligase kicks in. There is some more biology in there. There is a paper which I've linked in the article, which I'd really encourage you to read if you want to really dive into that. With regards to the dose, the clinical trial results show that it is a lot cleaner than the kind of standard of care PI3K kind of wild type inhibitors. So compared to alpelisib, which is the ones used, they saw they saw only a grade 3 to 4 hypoglycemia of about 6% versus 37% of alpelisib. They put a lot of effort into making sure this was selective, so I don't think we can say that it's pan. But yeah, again, we link the phase 2 results and then the press release. So again, if you really want to kind of dive into that, a lot more information is there. But I think it is genuinely a lot cleaner because of this novel mode of action where it's not engaging the wild type and it's only really engaging it in these tumor tissues.
Yeah, thank you, Matt, for elaborating on that a little bit more. Then, yeah, we also have one question for Rory here from Earth. So excellent overview. Any guidance on how to impact the liver to plasma ratio, for example, in the resimetirom case?
Yeah, it's an excellent question. I'm actually just doing a deep dive on this right now because I saw this one come in. Let's see if I can
Maybe I can speak to this a little bit in the meantime, Rory. So typically you will find many drugs that are enriched in the liver. That's actually not a very uncommon thing to see. If you think about, the different organs in the body after oral dosing, liver is actually the most enriched organ, and as you can expect, brain is the most impaired organ. So it's not super atypical to like see high kind of liver concentrations. But yeah, particularly this is an interesting aspect for liver diseases. And as you will know in the infectious disease space, of course, hepatitis B and C, oftentimes those drugs have also really high liver to plasma ratios. Maybe Rory can speak a little bit more in detail how it was thought about in the resimetirom case, but yeah, that's just a general overview that it's not something that is extremely uncommon.
Yeah, so they do have the JMC article, I think it came out in 2011 or so, where they just, make note of the, this distribution 8-to-1 liver to plasma. Sorry, it came out in 2014. But they don't comment on the, how to optimize for that, or if they were looking to, to get that preferential distribution into the liver.
Yeah.
I can also jump in
Yeah,
there.
absolutely.
One of the techniques that is used is if you can make it a substrate of one of the transporters that actively transports into the liver, that could be a really powerful technique. Lou and I were just at the Steamboat Springs conference where we saw the Chinook Therapeutics presentation on their lactate dehydrogenase inhibitor, where their approach was to have a very low passive permeability, but very high substrate selectivity for OAT1B or OAT1B3. So actually, you then don't get distribution into other tissues because you're low passive permeability, but you get active uptake into the liver. This doesn't come, engaging these can lead to other problems, but this can be really powerful effect if you have a very need for a very liver-specific mode of action.
And that's why for a lot of these liver targeting drugs, they have functionalities that are recognition elements for the uptake transporters like OATPs and things like that, like in that Chinook program, or bioisosteres of carboxylic acids, for example. One fun fact about xanomeline that we stumbled on in a preclinical study, I think in rats, they found that 10% of the dose wound up being in the brain. So sometimes molecules, lipophilic molecules like to stick in the brain. So that's another tissue you can target in different ways.
I can say, Brett, weren't you at Metabasis for many years?
So another approach is prodrugs, which are activated by P450 enzymes, which are concentrated in the liver. So yeah, that's what the Metabasis approach. That drug that you have that's in competition for MASH NASH from Viking is from Metabasis, and there's a prodrug, ester prodrug around that phosphonate that gets unraveled by P450 work in the liver, and that localizes that molecule in the liver. So I think that's the hope for the MASH NASH application. And I know people who are dumping money into the Viking stock because they believe that this is going to work better than what the one that just got approved.
Yeah, and I guess just one other thought is, of course, optimizing for low clearance and high stability in human hepatocytes is obviously very key. So.
All right. With that, yeah, actually Irv says thanks for the great answer. So thank you everybody for contributing to answering this. I think it's a good time, since we're on time right now, to close it here. I want to thank all the speakers for really excellent presentations and a wonderful overview over the last year's drug approvals. I want to thank the engaged audience for all your questions. Keep them coming. We're very excited to hear from you. And we want to leave you by saying, dial in next week. So next week we will have Alexei Garasudo here, and he's going to be talking about practical applications of physics-based modeling for medicinal chemists. He's the VP at Schrödinger and the head of medicinal chemistry there. So it'll be a very exciting talk, and you'll get to see how Schrödinger's methodologies are being used to optimize KPUU, for example, or basically predict KPUU a priori, which I think is a very difficult task, and learn more about some of the programs that they're working on. So with that, happy Friday, everybody, and we hope to see you next week or at any other Drug Hunter event in the future. Have a great weekend.